Hi, speakers, just a hold, please. We're waiting on some streaming issues, but you'll get the cue in the chat.
If it's actually moving for you.
All right, we are live.
Yeah.
Hello. Hello. Yeah, welcome to the symposium of relation of the brain networks. Yang from National Institute of Health. Together with me are Danny Wang and Colleen, Hanlon. The fourth speaker, Wolf-Julian Neumann, won't be able to the symposium today due to some issues. So we will have only 3 speakers in this session. Yeah, this symposium, yeah, we believe we will cover the cutting edge advances in neuromodulation of disrupted brain nets, and including the invention of mechanisms brain stimulation, concurrent neuromodulation, neuroimaging techniques. And we are also talk about the development of novel network-based approaches to effectively target the brain nets. Finally, we will talk about the involvement of treatment efficacy by imaging-based individualized optimization. So the format of this symposium, the speakers will summarize their talks first, and then followed by the Q&A sessions. So students can put your questions into the which is the upper right side of your Sparkle. So we will try to address your questions in a later stage. So, all right. So without further ado, I would like Colleen Hanlon to have a summary of her talks. Colleen, please.
Great, thank you very much, Yihong. I appreciate that. Just share my screen. Can you all see that okay?
Yes.
Excellent, great. Well, thank you all for joining us. I know it's varying time zones for those of you listening. As Yang mentioned, all of our talks have actually been pre-recorded. Today, we're just kind of giving a brief little summary. So this is a truncated version of my slides, but if any of you have any questions afterwards, I'd love to entertain them. Feel free to email me at any time if you can't stay for the end. So what I'm going to talk about to you today is some really new information we have about the importance of brain structure as a dosing technique for TMS. And so when we think about, I work on the addiction system a lot, but in fact, addiction is really just one example of something that's, I would argue, is sort of a fundamental process of being human. And this is this sort of duality between executive control and impulsive action. Those things go hand in hand. So when you have, when you're tempted to do something, you have this impulsive system, which is in the brain, much more medial and ventral, both in the cortical and in the subcortical, ventral medial striatum. But then you also have executive control that kind of weighs the other options. And so kind of pushes you towards doing another strategy perhaps, and not indulging in the impulsive action. So those are two ideas. And in our lab, we've been studying this sort of decreasing of cue reactivity as our target for developing TMS as a treatment for addiction. And so the reason that we chose that is because we did a neuroimaging study of over 100 individuals, 156 that came into our lab, and they had all received a basic block design cue reactivity task, which is somewhat standard in our field, where we showed them images of their drug of choice versus a neutral condition or a neutral beverage. And then we looked at what areas of the brain were active. And we found in this case that across many different disorders, smoking, alcohol, and cocaine users, there was this common transdiagnostically relevant activity in the medial prefrontal cortex. You also see there's sort of two little magenta dots on the outside that represents inferior frontal gyrus, or perhaps the anterior insula, which some of you may know that it was actually the insula was recently FDA cleared as a target, FDA approved target for treating smoking cessation with TMS. So our first target engagement studies, we wanted to see if we could even reach the frontal pole. And so we had a group of alcohol users and cocaine users. We applied TMS to the medial prefrontal cortex, real and sham. First, we put them in the MRI scanner, and we looked at their brain reactivity to drug cues versus neutral cues. In this case, we did a PPI analysis using functional connectivity, seeding the medial prefrontal cortex, which was our TMS target. And we looked at functional connectivity to cues versus neutral. And in both the cocaine users, there were 25 there, and in the alcohol users, 25 there was high baseline functional connectivity to drug cues. And that's something that's really common throughout the field. So we were happy about that. But then following one session of real versus one session of sham TMS, in both cases, the individuals had a decrease in functional connectivity between the medial prefrontal cortex and the striatum. And so this is we call this a sandwich design. So imaging, TMS, imaging, all in the same day. And it helps us draw conclusions a little closer to causality. So this case, we see that TMS, this is continuous theta burst stimulation applied to the medial prefrontal cortex, can decrease functional connectivity to drug cues in both cocaine users and in alcohol users. So that set up a multi-day clinical trial, which we ran. In this case, individuals came into the lab. They were treatment-seeking individuals. We did one with alcohol use disorder and with cocaine use disorder. They came in, they wanted to receive treatment. We gave them a baseline scan where we looked at functional connectivity to cues, and then we delivered 10 days of TMS to the medial prefrontal cortex. We then looked at them again at 1 month, 2 month, and 3 month follow-up. The actual full consort diagram of that study is in the full presentation if you want to watch that. With any luck, we'll get that paper published sometime this year, and I'm happy to tell you more. But just kind of the brief summary of the clinical outcomes, you can see here in on the left figure, the individuals that received sham TMS dropped out at a higher rate than those that received real TMS. So the enrollment, the odds ratio of maintaining enrollment in the program was almost 3 times higher in the group that got real TMS. And in terms of return to heavy drinking, the individuals in the sham group, which is the light gray line, returned to heavy drinking almost 2.4 times more often than individuals in the real group. So we were happy with this. It sets up some good clinical efficacy. When we looked at the brain data, as many of you all probably in the audience have done before, we have this sort of longitudinal fMRI design. We're going to do functional connectivity associated with this medial prefrontal cortex target. Here, our ROI is abbreviated FP1 to stand for the frontal pole. It's kind of this area right here. These are based on EEG coordinates. And in the group that received sham TMS, you can see that there was actually an increase in functional connectivity to cues relative to baseline at the 2-month and the 3-month follow-up. But in the group that received real TMS on the bottom, there was a specific decrease in functional connectivity. So that's all great, but this symposium is actually about sort of refining our ideas and what we can learn by sort of looking deeper into individual variability and how imaging can help inform treatment strategies. And so we thought, though this is great, we kind of took this data-based average, we did a computational model based upon this 156 individuals in this parent study, what place on the scalp surface would be most likely to capture the hot spot in the most number of people. But then when we actually look at individual data, individuals are really messy. And so when we look at individual data, we find that sort of the peak hotspot or the area of peak activity for each individual is kind of all over the place. So there's 2 things that could be responsible for variance. And this is work that's being done by Dan McCallie, who's a 5th-year graduate student in my lab, hopefully postdoc soon. And Dan has been looking at scalp to cortex as a distance as a factor that influences variability, as well as functional brain response to cues itself. And just kind of as a first pass, we did electric field models on each person's head. So when we place the TMS coil over the frontal pole, defined by EEG characteristics, what is the induced field at the level of the cortex? So if you think back to physics and Maxwell's equations, the intensity of a magnetic field or an electrical field induced by a magnetic field will decrease very, very quickly with distance. And one of the challenges of the frontal pole is that there's a fair amount of heterogeneity in each individual's cortical geometry. If you can see one another on the WebEx terminal at the moment, you can probably look around at your fellow colleagues and see everybody's head is really kind of shaped pretty different. But the brain inside of the skull is actually fairly uniform. So we have to get over this distance. And as you can see here, this is the electric field model for everyone involved in our study. And there's actually a decent amount of variance. Additionally, when we look at individual bold reactivity to alcohol cues, in this case, there's also a fair amount of variance. And so this is a cross section of the area of peak activity for each individual enrolled in the sample that had elevated activity to brain cues. And so you can see there's 9, 12, 15, 16, 18, 19 individuals shown on the left. And then we also had 18 individuals that didn't have significant elevated functional reactivity to cues at all. So frankly, 50% of our group wasn't even really cue reactive. And so if we think about that, maybe it doesn't make sense to put TMS over the frontal pole when there's not cue-induced activity in the frontal pole. So what Dan very smartly did is he tested this hypothesis that individuals where the brain map, so the contrast map, created in reactivity to drug cues versus neutral cues overlapped with a TMS-induced electric field would have the best response to TMS, right? So in this case, the brain, there's bold signal there and the TMS electric field reaches there, those people would do the best. He called those TMS candidates for the sake of this talk. And you'll see them here. So in the purple line, he has the best TMS candidates with overlap, those that did not have overlap, and then the sham group. And in our individual sample, if you look at the percent of the sample that was had at least one heavy drinking day in the last 30 days, at baseline, all three groups are about the same. After 10 days of treatment, though, they start to separate. At 1 month, they're still separated as well as 2 months. And so as you can see here, actually the purple group, the group with highest overlap between the TMS-induced electric field and the bold signal were responded the best in terms of alcohol drinking. So I think that's actually really exciting. And what we learned is that it's actually not just electric field alone. I didn't show these data, but we looked at each individual source of data independently. And we found that independently, these two things don't account for variance in treatment response. But when you put these two things together, you can, they're highly correlated with individuals that will actually respond well to the treatment and remain sober. And so the next step in our line of research is to see if we can actually prospectively predict who's going to respond really well and potentially refine the targeting. And that's all I have. Thank you.
Yeah, great, Colleen, thanks. So yeah, just for a reminder, audience, if you have a question, so you can put it in the chats and we will address your questions in the end. So next, I guess I would give a brief summary of my talk. So let's see. Yeah, looks okay, the slides.
Yes, look great.
Okay, great. So I'm going to talk about a computational framework which can be used to target the pathological network. So this slide just shows the concept of this method. So on the left side, we can think about we use TMS, for example, to stimulate one side of the brain, but this stimulation will generate the whole brain effects. So which could be excitatory in some region or inhibitory in other regions, as a kind of schematically showing on this matrix. So then on the right side, the panel B, we in the center, there's another network called the pathological network. So this can be formulated in several different ways. But one way is to look at the brain activity between the patient cohort and healthy control cohorts, for example. Then the ideal modulation would be, this regulatory network would match to this pathological network, or a kind of counter intact with this pathological network. So this will be generate more effective treatment effects. So compared to some others, for example, untargeted treatment, they don't match each other, so it would be less effective treatment. So yeah, so this is basically a concept, and so basically there are two networks. One network is, as I mentioned, a pathological network, which can be derived from the brain activity difference from patients and controls. Another is we call a modulatory network. For example, it's a TMS-induced effects from stimulation sites to the whole brain through the connectivity. So then the goal is to match these two networks so that they will generate the maximum treatment outcome. So just give you an example. For example, here we look at the TMS stimulation just within the dorsolateral prefrontal cortex, and but slightly different locations. So then we can calculate the modulatory effects based on functional connectivity, for example. Then the pathological network, again, can be derived from the brain activity difference between the patients and the controls. So then just between comparing these two networks, and we can estimate the network targeting accuracy as shown here. So for different stimulation site would generate a different targeting accuracy. So that's basically the concept. Then we test this model using several clinical data sets. So again, the pathological network here is generated from a brain activation, actually acquired from a PET study, and the functional connectivity. The modulatory network is generated by functional connectivity from a large cohort of subjects. So then we test this on the first data set, which is a TMS on the 5 centimeter anterior to the lateral motor cortex, as shown here. So based on each subject's TMS location, we can estimate the treatment outcome and then compare to the real treatment outcome. So yeah, so they are significantly correlated. In the second data set, and we again look at the TMS treatment based on the hypometabolic frontal regions. So this is basically from a PET study to generate the metabolic map, and for each subject, look at the decreased metabolic rates compared to controls. Then you can see the stimulation sites here just across the both left and the right side of the frontal cortex. So again, using our model, we can predict the treatment outcome significantly. So besides the previous two cases are from major depression. So besides that, we also look at the hallucination in schizophrenia patients, for example. Here, the pathological network is generated by the hallucination on versus off, right? And the modulatory network, again, generated by a large cohort of subjects' functional activity. Then if you then we test this on a real data set. So this is a real treatment, TMS treatment on the response to a language task, particularly in the superior temporal gyrus. So as shown here, and then based on individual stimulation site, our model can predict the treatment outcome significantly. So besides, you can kind of validate this from retrospective data sets. We can also predict some new treatment. So you can imagine instead of predict the treatment from a single site, you can just move around this ROIs across the brain. Then you can generate a map of this treatment accuracy. So if you put some threshold, for example, the highest 5%, you can see those regions would have best treatment effects. So including the bilateral dorsolateral prefrontal cortex, which has been widely used in the treatment of depression. And also, interestingly, you can see some other regions like anterior insular and anterior cingulate cortex, medial prefrontal cortex, and the PCC regions. Those regions haven't been widely used in the treatment of depression yet. But from the model, and we can predict those regions would have very high treatment efficacy. So yeah, just want to just summarize. And we kind of developed a computational model for network targeting that quantifies the treatment effects of the TMS. This model being validated through retrospective data sets, both in major depression and the audio-video hallucination. suggesting this model can be used in the transdiagnostic disease models. And finally, I think very interesting is that this model can be used to predict new stimulation sites with potential high treatment efficacy. And this will guide the future treatment experiments. Yeah, that's all I have. Thank you very much. Yeah. Next, yeah, I would like to have Danny Wang to have a summary of his talk. Again, so for audience, if you have questions, just please put into the chat box in the Sparkle. Daniel, please.
Okay, yeah. Can you see my screen and hear me?
Yes.
Okay. Okay, great. Yeah. So I'm very excited to hear the first two talks and continue this theme. My talk was mostly on improving TDCS. So it would probably be the least accurate but least invasive. There has been exponential growth in TDCS during past decade. And also there's a lot of variability across subjects, across studies. So our goal is try to really improve the efficacy and accuracy of TDCS towards personalized TDCS treatment with neuroimaging. The technique we come up with is this dual echo EPI, which is quite a standard, in neuroimaging by other groups. We do this concurrently with TDCS, and you get both magnitude and the phase. The magnitude give you the bold signal, and if you do magnetization preparations such as arterial spin labeling, you get also a cerebral blood flow, CBF. And you can analyze this data, as Yihong mentioned, you can get functional connectivity as well as CBF. On the other hand, we can also map the magnetic field induced by TDCS with this magnetic field mapping of based on the phase of the signal. This phase magnetic field change is actually proportional to the applied current density, based on this Ampere's law. Of course, there's because it's TDCS, so we don't have this time dependent varying electric field. The way we do this experiment is very similar to an FMI experiment. You apply a block design of TDCS with different amplitude, and then you do this dual echo EPI scanning simultaneously, and you can then use general linear model to decompose, the magnetic field changes correlated with your applied current. You can also covary out, confounding factors such as head motion, field drifting. So we applied this technique in phantom as validation. This is a current carrying wire through a water field phantom. This is based on simulation, the magnetic field distribution. This is what we measured. This is when we flip the current, change the polarity, you get really the opposite map. And this is the sham condition. So we get very good correlation up to sensitivities up to nanotesla per milliamp current applied. We also did an in vivo experiment. This is a retest in 7 healthy subjects with bilateral motor cortex TDCS. So you will see this a very good retest repeatability with an average ICC of 0.8. And this is simulation using SimNIM software. So when you compare the in vivo results with the simulation, they are largely comparable with the correlation of 0.57, but there's some individual variability. So we can see. There's also a little bit caveat related to, directly the current causing the field changes and causing susceptibility effects, and there's both signal change caused by the magnetic field, and it's very adjacent to the wire, so that's good. And if we are away from the wire just a few centimeters, then we don't see any signal change. Then with the both signal, you can do network analysis, and this is retest results. We can detect the common networks, and then also the network that is under the cathode and anode electrodes that is correlated with both signal changes. Another technique, we have been using quite often is arterial spin labeling perfusion. This is a paper published by Dr. Michael Nish's group. Basically, this shows the following an anode stimulation on M1, you will see a persistent CBF increase. There's a persistent CBF decrease following cathode stimulation on M1. So with all these tools, we applied this technique in major depression. So there's actually quite a bit of publication in the clinical trials, major depression using TDCS. So these are the two major studies published recently, one positive, one negative. So there's some controversy. So the pilot study we've done is led by Dr. Catherine now at the UCLA. It's a 4-arm study with a conventional TDCS and the HD TDCS and associated sham condition. So this is a 12 sessions, 2 weeks. And the neuroimaging was performed before and after. This is the montage HD 4x1. This is conventional. The location was verified at visit 6 mid-trial with neural navigation. The imaging technique we use is 3D Picasso for CBF and the dual echo EPI for magnetic field mapping on the board. This is the results published this year. So this is the HD showing the magnetic field. Basically, it has a side flip under the electrodes. Basically means you can intuitively imagine a current injection along this line. For conventional, as expected, you will see a diffuse kind of distribution of magnetic field in frontal. And this is sham. So for the magnetic field, the effect size is very similar with conventional and HD. And there's a cluster under the electrode also correlated with both signal change. For ASL, the two main clusters we observe is this is prefrontal, left dorsolateral prefrontal cortex, which is the target area. Another is ACC. So for this one, it's very interesting. The ACC was only significant with HD, not with the sham, with the conventional. And if we compare HD versus conventional, we actually detect subgenual ACC activation. So behavioral, because it's a small trial, there is no significant change in Hamilton score, but we do see significant SHAP score decrease with HD, but not with conventional. So overall, this study seems to suggest, the HD has better focality as well as, effects. I mean, the network connectivity with ACC as well as behavioral effects indicated by the SHAPS. So there's an ongoing study evaluating HDTD cells in MDD. Would like to thank my colleagues at USC and especially UCLA. Most of the work was done by Mike Jog from a postdoc in my lab, as well as my colleagues in CUNY. Thank you.
Yeah, great, Annie. So again, if audience into the chat of the Sparkle is on the upper right side of the platform. So yeah, I think while the questions, I just have a kind of curious from a Colleen study. Colleen, you mentioned some acute TMS, like just a single session, your sandwich kind of treatment. So kind of curious about the relationship between the acute TMS effects. Would you comment on that based on your experience?
Yeah, just to clarify, you broke up a little bit. Do you mean the effects on the brain or on behavior?
If that's brain, yeah. Or maybe both.
Yeah, great. So we from a brain perspective, we actually saw very similar effects in some ways. So the acute TMS was a session of, so you're in the scanner and then we get tapped on your head and then we put you in the scanner again. And we brought those individuals back again about 2 weeks later. We did the same thing, but giving them either real or sham, whichever they didn't have. And so whereas in the multi-day trial, we actually did a scan while they were receiving behavioral therapy and TMS, and then another scan and a 1 and a 2 month follow-up. So in some ways, the general actually pattern is very similar, that real TMS decreases functional connectivity to alcohol cues between the medial prefrontal cortex and particularly the caudate, putamen, and the insula. So those things seem to hold up in both cases. But as you guys know, behaviorally, one session of TMS doesn't really do anything, right? It probably changes cortical excitability. Well, I guess it can change cortical excitability. It might change subjective craving for drugs, although that's highly variable. But what most interestingly when you give many sessions of TMS, and this is what like they would do in depression, they give 30 sessions or 36 sessions. When in our case, we gave 10 sessions of a really high potency form of TMS. It was 3,600 pulses of continuous theta burst. In that case, we actually were able to improve relapse to alcohol use. So I think you definitely need, the brain imaging is fairly consistent, but you need the power of many sessions in order to get some type of behavioral change.
Mm-hmm. Yeah, no, that's interesting. So if the acute can generate a kind of brain response similar to the chronic, would be able to use the acute guide for look at the inferences or some planning for the treatment.
Yeah, that would be awesome, right? I think I have heard that, I don't know that anybody's done that, but that would be great. I have heard that actually there's some predictive validity in using in depression, using their behavioral response to the first treatment of TMS as a good indicator of whether or not they're going to stay enrolled and complete the treatment. So yeah, I think that would be a great area to look into for sure.
Yeah.
So
thanks, Colleen.
Yeah. how far you think we're really away from like a really personalized modulation, like a n equals 1? You look like you guys are very close to implement that.
Yeah, yeah, we actually we have some TMS and MI, just like you know, in Colleen's group in probably they also have that in South Carolina. So what are we trying to do is using the TMS MRI to look at the acute effects, then use this acute effects to predict the brain effects to predict the chronic, just as I mentioned, asked Colleen about. But we haven't really done that yet. It's in our plan. But yeah, I know Colleen has much more experience on the real TMS treatment, you know, protocols. So yes, yeah, I hope you know, that will be a way to kind of to be more personalized treatment. Yeah.
You know, I'm in a, I think Danny, it's a great question. I would say, so I'm in a cancer biology department currently, which is a sort of a new place for me. But what I've learned a lot is that the oncology world is actually very, very close to n equals 1 type treatment for cancer. And they seem to have done so by combining heavy genetic markers as well as sort of pharmacotherapy and patient tolerance and behavioral attributes. So I think I would love to say that we're getting closer. But, you know, outside of oncology, I think it's, you know, it's really hard to have n equals 1 treatment for anything. But certainly it's exciting. And you know, the data that Dan McCallie, my graduate student, came up with, I think is kind of moving in that direction. I think there's other people using EEG as potentially like a predictive biomarker, which to me, that's really attractive because EEG is really scalable, right? So you could deliver EEG, you know, to rural communities and things a little easier than fMRI. But Danny, that's actually what I love about TDCS as an approach. I think TMS, you know, it is used more clinically, but in fact, it's really not a great treatment. Like in terms of COVID times, we're very much aware that, you know, people that live in rural communities, there's this disparity in like their access to care and their ability to come. So TDCS opened up a huge window of opportunity there, which I think the stuff you're doing is really important.
And I mean, that's an eventual goal. I think that's why people are enthusiastic about it. But yeah, there's still a lot of obstacles. Even with home treatment, you know, there's also I think, you know, especially with the MI guided and neuroimaging guided. So we're facing the same barrier as you're facing. And TMS, I think, in a sense, from a regulatory perspective, it's a medical treatment. I think it's better regulated. Yeah. So yeah, I think each has some pros and cons.
Yeah, Danny, you mentioned that there's a high density TDCS compared to more conventional TDCS, and I think you showed some interesting results, like high density would generate a kind of more blood flow change in subgenual ACC. That seems to that is very amazing. So how deep the high density can kind of generate, or maybe how focus of the high density would be compared to the more conventional. Yeah.
Yeah, so there's actually many papers on that. By the CUNY group, Aaron Dickinson and his colleagues. So they invented HDTD sensor. So it's 4x1. So with one anode injecting, but it returns in four kind of circular fashion cathode electrodes. So they have many papers, with computational modeling. I think the accuracy is generally on the order of centimeter, a few centimeter, depends on how far you put away the cathode and the anode. And we have done, we're probably the first group that done in vivo, like a magnetic field mapping with this technique. So I showed one slide, it's still not very focal, but it's better than conventional. I think I can share again. Yeah, so like this one, it's, you can see the flip from positive to negative field. So in general, we discussed last time about from the B field to the E field. It's still challenging to directly derive the E field, but you can see it's more focal towards the prefrontal cortex, while the standard you get really diffuse distribution. You're sweeping through the whole prefrontal cortex. So that's pretty much right now what's available right now. But there's actually more configuration, because people are coming out with HD, not only 1x4. Basically, now the EEG cap, you can reverse it, right? In a sense, even people doing inverse EEG. Basically, if you know a source, you have a distribution, then you can theoretically, if you inject the same amount of current into the electrodes, you will get a reverse kind of. So there's actually a company coming out with multi-channel, both EEG and TES, transcranial electric stimulation. So the field is still evolving. I think, that in that sense, I think Colleen is right. There's portable EEG, multi-channel EEG system. They're both monitoring as well as manipulating brain electric activity. So it's a very exciting field.
Yeah, great. Thanks, Danny. Yeah, I think it's it's up to the time, and we have to conclude. And I'd like to thank the speakers, Danny and Colleen. Thanks a lot. And I also like to thank the audience for attending this session. And that's it. Hopefully, see you next year. All right. Yeah.
Okay. Yeah.
Okay. Yeah.
Thank you.
Bye bye.